Okay. So, anybody removed outside? I don't think so. Today I'll be talking about somatic cell hybridization. Somatic cell hybridization essentially means two somatic cells, that is, two cells which are not sperm or egg. They fuse and they produce a new type of cell. So essentially, this is a very important topic in plant and botany, plant science and botany, where this particular technique is used extensively. In animals, something similar happens, and that is known as hybridoma technology. It's something different. So I'll start to share my screens. First, I have to open them. Just a few seconds. I'll just get it ready. It must be visible now. Are you able to see this?
Yes, sir.
Okay. So today's topic is somatic hybridization. Essentially, you can see that you isolate cells from these are two this is plant 1 and this is plant 2. So what you are doing is you're isolating one cell from here and isolating one cell from here. Then through certain means you are fusing them together. And then you are selecting them, and that particular cell is made to grow into a new type of third type of somatically fused cell. So you fuse one cell from here and one cell from here, this plant. This is two different species, and you have a third new type of plant coming. This is known as somatic hybridization, and this is done extensively. So essentially, it can be defined as the development of hybrid plants through the fusion of somatic protoplasts of two different plant species. And now I will explain what is protoplast. You take a leaf cell or any plant cell and you digest the cell wall off. If you digest the cell wall off, you have the cell membrane and the whole cell is remaining. And that is known as the protoplast. In plant, it is as it's a very handy tool because these protoplast can be transformed with DNA, and you can see certain you can do lots of reporter assays, and lots of things can be done. You just remove the cell wall, which is a very impermeable barrier, and then you have it almost like an animal cell. Of course, it has got chloroplasts inside. So the technique is, first step is you have to isolate the protoplast. Then the next step is fuse the protoplast of the isolate protoplast of two different species. Then you have to fuse the protoplast of those two different species. Then you have to identify which of these cells have actually hybridized. They are fused together, and then you culture these hybrid cells. And then from there, with the help of hormone treatment, you can regenerate hybrid plants. So these are the 5 major steps of somatic hybridization technique. So now this is how a protoplast look like. Are you able to see this picture of protoplast here? Basically, they are round. দেখতে পাচ্ছ?
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
I'll try to see if in the internet I can get better pictures of protoplast. I used to work extensively on protoplast during my PhD. Lots of things. So essentially, what you do is technique of protoplast. So were you people able to hear this?
No, sir, no audio.
No audio. Okay. There was no audio. Okay. Anyway, it should be there in the recording. So many people are entering so late. So let's try this once more. Okay. So this is how actually the protoplasts look like. This is how the protoplasts look like. You have digested the cell wall essentially. You have digested the cell wall with enzymes known as masarozyme and cellulase, and then you get these little round protoplasts coming, and they are used extensively for your studies. Just 1 second, I will share my screen once more. Because of entry at late point, somehow it disturbed. Okay. So let's get back. So these are the protoplasts which are formed. So the protoplasts, also known as naked plant cells, because they don't have the cellulose covering, cell wall cellulose covering. And it refers to all the components of the cell, excluding the cell wall. Apart from the cell wall, the other things are all there. Protoplast is biologically active and the most significant material of the cell. Then plant cell wall, actually it acts as a physical barrier and protects cytoplasm from microbial invasion and environmental stress. We know that. And it is a mixture of cellulose, hemicellulose, pectin, lignin. এরপরে আর ঢুকতে দেব না। Cellulose, pectin, lignin, lipids and proteins. And so to dissolve the components of the cell wall, you have to have the respective enzymes present. So the protoplast term was given by Hanstein, and isolation of protoplasts from plasmolysis was achieved by Clarker, and Cooking in 1960 isolated protoplast of plant tissues by using cell wall, in which we use a lot: cellulase, hemicellulase, pectinase, and protease extracted from fungus. And first protoplast fusion was done by Power in 1970. And protoplast is isolated. Basically, you know that it can be done by mechanical method, or it can be done by enzymatic method. Mechanical method, the mm, plant tissue is placed in plasmolysis solution, so where the cells plasmolyze. That means the cells, they shrink away from the plasma, from the cell wall. So and then you observe the cell, and then you cut the cell wall with knives. So you just chop, and then in this particular solution you release protoplasm and you collect the protoplasm. And it is used for vacuolated cells like onion bulb scale, radish, beetroot tissues. You get a very low yield of protoplast. It is very laborious and tedious process, and there is low protoplast viability. So this method is not preferred. Best is, which we always use, is the enzymatic method. You grow the leaves in a sterile chamber, in covered containers where light and everything is present. There is a membrane filter attached where air and all things can enter, carbon dioxide can enter, and you put them in a plasmolysis solution, and you give pectinase and cellulase. So you digest it, and then you isolate it, and you can isolate it. After you digest the cell wall, you get very good quality protoplasm. So this is used for a variety of tissues and organs, including leaves, petioles, fruits, hypocotyl. Normally, leaves, leaf tissue is the most suitable source. I used to use Nicotiana plumbagifolium, and that is tobacco plants. And you have high yield of protoplast, and it's very easy to perform. More protoplasts survive. There is a lot of death, but then you separate the dead protoplast and the living protoplast. Now we come to the protoplast fusion part. Fusion of protoplast by two different genomes. So one may be one plant species, another is one another plant species. These you can fuse them spontaneously, and this can be intraspecific or intergenic. But mostly what it is used, protoplasts are fused through induced fusion. It can be chemofusion, mechanical fusion, electrofusion, these three types. Normally, chemofusion is the best preferred method. So spontaneous fusion happens when you are isolating protoplast, They suddenly, uh, the same protoplast, they fuse and produce homokaryons. So plant species one protoplast plus another plant species one protoplast, they both fuse and produce a new type of protoplast, but it contains the same species, becomes more like a polyploid. That is known as homokaryons. They have no, it is between the same species or genera, and it has got no importance. This happens accidentally, this spontaneous fusion. What we need is the intraspecific protoplast fusion. They tried to make protoplast fusion between tomato and potato, where you will have the roots will be like a potato, so you can, and you will have fruits like a tomato. The name was pomato. Unfortunately, they did it the technique of protoplast fusion, but they got the other way. They got the roots of a tomato and the fruits of a potato, and therefore it was a flop experiment. But still, people are trying different ways and combinations of making this. That in one plant you will get both potato and tomato. They all, both of them belong to Solanaceae, and the fruits of a potato, they look like tomato, but they are a bit pinkish and small, and they are not actually healthy for human consumptions. Even cows avoid eating them. They have got certain toxins. Solanaceae is a toxin family, but that of tomato, it is not a problem. So intraspecific protoplast fusion is the cross between the same species in an individual, which involves isogenic strains or the non-isogenic ones. The true value of protoplast fusion as means for establishing crosses has been realized in few fungi. In fungi also this has been done in Cephalosporium. This technique offers the only way of carrying out crosses and genetic analysis in many fungi. So this protoplast fusion technique, it made it possible to produce preliminary genetic map of 8 linkage groups of Cephalosporium. Now let us talk about interspecific protoplast fusion. This fungi, it was essentially two crosses were done, like you did a Drosophila 3-point cross, and you found the gene map and linkage map. Here, by this protoplast fusion technology, it's like a cross. You are making, you can make a genetic map also. The frequency with which two characters can come together. This was used in this particular technique. So now we talk about the interspecific protoplast fusion. Interspecific protoplast fusion is the crosses between two different species. Interspecific protoplast fusions are of much importance in the area where new products are to be produced. Due to new genetic setup, many novel secondary metabolites such as antibiotics may be produced. Some of the examples where interspecific hybrids were produced are in yeast. Then in the two different species of yeast. Now, induced fusion, this is what we do. What you use is polyethylene glycol, fusogens, they fuse two cells, polyvinyl alcohol, calcium ions, sodium nitrate, NaNO3. But PEG is the most preferred one. It fuses the cell membranes. Fusion can be mechanical fusion, physical forms of protoplasts under microscope by using micromanipulator and perfusion micro pipette. Normally we do use this one. You can even use electric current, and you can do this with this fusion. So the protoplast fusion and the somatic hybrids which are formed, the fusion process, it can be electrofusion. The protoplasts are aligned in a special chamber. Electric current is applied, opening channels in cell membrane. So the electric current, it open channels, and once it open channels, they can fuse. Material from one cell can go inside the other cell. In PEG fusion, they are coated with PEG and incubated together, and they cause the cells to fuse. After fusion, the fusion products, they are actually round. After fusion, they become a bigger round. So this is the picture. One of them is fusing with another one in tobacco plant protoplasts. They are fusing, and a big round protoplast has been made. They have been fused. The next thing is to see if the protoplasts after fusion are viable. So they use this Fda staining to see if cells are alive. Or these type of cells they are used to see that they are viable or not. If they stain, then they are dead. So Fda is a dye that accumulates inside the plasma membrane of viable protoplasts. Viable intact protoplasts fluoresce yellow-green within 5 minutes. So different stainings are done. Then the next is you check the protoplast density. Protoplasts have both maximum as well as minimum plating density for growth. Now you put them on plates to grow. Then the next thing is you select. For this selection, I will take another slide. It's much better in that. Okay. So now selection of so two protoplasts fuse and they form heterokaryon. Now selection of hybrid cells. So about 20 to 25% of the protoplasts are actually involved in the fusion. After the fusion process, the protoplast population consists of a heterogeneous mixture of unfused chloroplasts, homokaryons, and heterokaryons. So, strain A, strain B, the protoplast fuse, you want this heterokaryon. But it could well be that there is a spontaneous fusion. A can fuse 2 times and form A plus and form a homokaryon A plus A, or there can be a homokaryon B plus B. Now you have to identify them. It is therefore necessary to select hybrid cells, heterokaryons. The commonly used method employed for the selection of hybrid cells are biochemical, visual, and cytometric methods. So it is the identification of hybrid cells. The development of hybrid cells followed by the generation of hybrid plants requires a clear proof of genetic contribution from both the parental protoplasts. So chromosomes from both the parental protoplast must be coming inside. So first is the molecular technique. Through PCR, you have markers for strain 1, markers for strain 2, and you do a PCR. Marker is nothing but a particular specific gene for strain A and a specific gene for strain B. So by PCR analysis, you can identify this particular two of these genes, and you can say, okay, very well. Then you can see the restriction enzymes are enzymes which digest DNA at specific points. So different types of DNA have different uh, digestion patterns. So you can identify these restriction patterns uh, through these molecular biology techniques. Then you can count the chromosome numbers. So strain A has got 7, strain B has got 8. So the hybrid will have 8 plus 7, 15. So you can count the number of chromosomes and then you can say, okay, this is a hybrid. So these are the ways by which you can actually screen for protoplast fusion. The next stage is the regeneration of hybrid plants. So you have screened for the hybrid and now your regeneration. They are induced to regenerate from hybrid callus. So in a media, they are given hormone and these one protoplast, they grow cell wall again and then they become a callus, a sort of tumor-like structure. And they must be partially fertile. And then they are grown into plants. So this pomato was a hybrid of potato and tomato was done, but this failed. But you could have a better, you could have had a better result from this. So this is the process of somatic hybridization. So do you people have questions? কোনো প্রশ্ন থাকলে জিজ্ঞেস করো। আছে প্রশ্ন কোনো?
No, sir.
ওকে 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 দেন থ্যাংক ইউ। আজকে থিওরিটিক্যাল ক্লাস করতে পারবো না, ওই সময় আমার একটা ব্লাড টেস্ট আছে, আমার ভাইরাল হয়েছিল তো তো এখন ওরা সুগারটা উল্টো পাল্টা হয়ে যায়, তাই জন্য ওরা এই 10টার সময় আমার একটা টেস্ট করবে। যদি উল্টো পাল্টা হয়ে গিয়ে থাকে তাহলে ওই আর কি আমাকে রেগুলেট করতে হবে। তো ঠিক আছে, বাই বাই।
Okay, sir. Bye, sir.
Thank you, sir.
ইউ ওয়েলকাম,
Thank you, sir.
ইউ ওয়েলকাম।